Dear students, in this video we can discussing about microbiological assay of antibiotics. Before starting the topic, let us have an idea about assays. Now, what is assay? As you know, assays are investigative procedures for qualitative assessing or quantitative measuring the presence or amount or the functional activity of target entity, which is always known as analyte, which can be a drug or biochemical substance or organic matters. So the assays are methods or procedures used for qualitative or quantitative measuring the presence or amount of the active ingredient or functional activity of the analyte. Okay, then the assays are mainly done by physical, chemical or biological methods. In biological assays, we are using different organisms or tissue parts or cell parts or body parts for determining the activity or the functional group of the testing analyte. Okay, then here we are discussing about the microbiological assay of antibiotics. That is, in microbiological assays, we are using microorganism for qualitative or quantitative determination of the chemicals, biochemicals or the tested analyte. Clear? Then what is microbiological assay? Microbiological assay is a powerful analytical technique that uses living microorganisms to determine the concentration or biological activity of chemical compound in single or complex material. That is, microbiological assays are a qualitative or quantitative determination of any chemical compound from a single or even complex material with the use of microorganism. It is performed to measure the relative potency or activity of substances like antibiotics, vitamins and amino acids by determining average response of a microbial culture. So, these microbiological assays are performed to measure the relative potency or activity of substances like antibiotics, vitamins and amino acids. Clear? In case of antibiotics, activity is estimated by measuring inhibition of microbial growth. And in case of amino acids and vitamins, activity is estimated by measuring the promotion of microbial growth. In case of antibiotics, the activity is estimated by measuring inhibition of microbial growth, and in case of amino acids and vitamins, the activity is estimated by measuring promotion of microbial growth. And the response produced by test substance is compared with that produced by non-concentration of standard preparation of that substance. And finally, the response produced by the test is compared with the standard, and this response produced by microbial culture is directly proportional to the concentration of a substance under test. Then what are the advantages of microbiological assay? The microbiological assays are simple, specific, inexpensive and convenient method which require minimal specialized equipment, and this produce accurate standardization of medicinal compounds for pharmaceutical quality control, and also these are ideal for assaying naturally occurring therapeutic agents with complex structures, and it also enables precise determination of biological activation of compounds in therapeutic context, and it is very useful for determining changes in antibiotic potency and preparation quality, and it is unique in application, and they produce rapid results compared to many other chemical analysis methods, and it also uses minimal sample amount and can test large number of compounds simultaneously. Now we are discussing about The demerits of microbiological assay. The first disadvantage of microbiological assay is it requires specific organism against the specific test material. And the second one is it needs sterile conditions throughout the procedures. And the next disadvantage is the slight temperature variations during the incubation procedures may vary in the result. And the next one is to require extended incubation periods, making it slower than some other chemical methods. And which also require well-trained individuals with specialized microbiological knowledge are essential for doing the microbiological assays. So, the demerits of microbiological assay including it requires specific organism, it requires sterile condition which are temperature sensitive, it is more time consuming than some other chemical methods, it need expertise individuals or trained individuals for doing the microbiological assays. Then we are discussing the needs of microbiological assays or why microbiological assays are essential. It is necessary to assay microbiological agents for determining the potency, pharmacokinetics of drug in animals or humans and for monitoring and controlling antimicrobial chemotherapy. Okay, so it is necessary to assay antimicrobial agents for determining potency, pharmacokinetics of drugs in animals or humans and for monitoring and controlling antimicrobial chemotherapy. So the needs of microbiological assays including first one the determination of potency, that is to establish a baseline effectiveness of antimicrobial agents, to establish a baseline effectiveness of antimicrobial agents. And second one is pharmacokinetic studies that it track drug behavior and concentration changes over time, that is it track drug behavior and concentration changes over time. And the third one is therapy monitoring, that control and optimize antimicrobial treatment protocols. So these are the needs of antimicrobial assay, that first one potency determination, second one pharmacokinetic studies, and third one controlling the therapeutic monitoring. Then what are the principles of antimicrobial assay or microbiological assay of antibiotic? The principle involves or the assay involves determining the potency of a test antibiotic to inhibit microbial growth and comparing the result against a standard antibiotic preparation whose potency has been established with reference to international standards. That is, in microbiological assays, it involves determining the potency of a test antibiotic to inhibit microbial growth and comparing the result against a standard antibiotic preparation whose potency has been established with reference to international standards. This comparison allows for precise qualification or quantification of antibiotic strength in the test sample. So the comparison of test and standard allows for precise quantification of antibiotic test in the test sample. The two main types of assays used for antibiotics including serial dilution method and second one is diffusion method. The first one is serial dilution method and the second one is diffusion method. In serial dilution method, we measure a minimum inhibitory and bactericidal concentration through progressive dilution of test. That is, in serial dilution method, we measure minimum inhibitory and bactericidal concentration through progressive dilution of test solution, and diffusion method to measure the zone of inhibition as antibiotic diffuses through the agar medium. That is, in diffusion method, we measure the zone of inhibition as the antibiotic diffuses through agar medium. So the first one is serial dilution method. The dilution of the antibiotic under test that will inhibit microbial growth is compared with the standard. In serial dilution method, the dilution of antibiotic under test that will inhibit microbial growth is compared with the standard. From the potency of the standard, the strength of unknown can be calculated. That is, in serial dilution method, we are determining the concentration of antibiotic under test that will inhibit the microbial growth and which is compared with the standard antibiotic solution. Okay. The steps involved in serial dilution methods are, first one is the preparation of serial dilution. So, we are creating series of antibiotic solutions in test tubes. And second one, we are inoculating standard bacterial suspension to each tube. The specific bacterial suspension to each tube which are already prepared with the series of antibiotic dilution. And the third step is analyzing or identify the inhibition points after incubation of the microorganism. So, in serial dilution method, we are preparing serial dilution of antibiotics in each test tube, and then we are inoculating standard preparation of specific microorganism for the test into each test tube. Then we are incubating it at specific conditions of temperature, and then we are analyzing the inhibition ability or inhibition potency of the test solution in each test tube. Okay, the key measurements including the serial dilution methods are minimum inhibitory concentration and minimum bactericidal concentration. And what is the minimum inhibitory concentration? The minimum inhibitory concentration is the lowest concentration or the highest dilution of antibiotic that inhibit visible bacterial growth. That is, the minimum inhibitory concentration is the lowest concentration or the highest dilution of antibiotic that inhibit visible bacterial growth. Okay. Then what is minimum bactericidal concentration? The minimum bactericidal concentration is the lowest concentration of antibiotic that kill bacteria. Clear? The minimum inhibitory concentration is the lowest concentration that inhibit the growth of bacteria, but the minimum bactericidal concentration or MBC is the lowest concentration of antibiotic that kill the bacteria or achieves minimum lethal concentration. Clear? Then the two types of dilution test including broth dilution and agar dilution. We have two types of dilution test. The first one is broth dilution test and the second one is agar dilution test. In broth dilution test, we are preparing series of antibiotic dilutions which are prepared in tube containing broth medium. So, we are adding antibiotic dilutions or antibiotic dilutions are prepared in tube containing nutrient broth medium. And in agar dilution method, we are using series of antibiotic dilutions prepared in agar medium which is poured into plate. So, we are preparing series of antibiotic dilutions in test tubes with agar medium, and when after adding the test organism and antibiotic dilution, we are transferring it into or pour it into agar plates and then allowing the agar plates to solidify and then it is incubated. Okay, in case of broth dilution method, we are using liquid medium, and in case of agar dilution method, we are using solid agar medium. Clear? So, in case of broth dilution method, the microorganism grow in the liquid medium and it is visible as the turbidity. But in case of agar dilution medium, the microorganism grow in the agar medium, so we can visible the growth of microorganism as the colonies. Okay, the colonies of microorganisms. If there is no visibility of turbidity, then it indicates the antibiotic inhibit the growth of bacteria. In case of agar dilution test, the absence of the colonies indicate the antibiotic kill or inhibit the growth of microorganism. As we already discussed, the serial dilution method include two methods, broth dilution and agar dilution method. In case of broth dilution method, we are using series of test tube which containing nutrient broth medium, and we are preparing the antibiotic dilution in the nutrient broth medium. Clear? So, the series of test tube containing nutrient broth medium, and we are preparing the antibiotics in this nutrient broth medium. Series of dilutions of antibiotics in nutrient broth medium. Okay, so this test tube containing 64 microgram per ml of antibiotic. Then the second test tube containing 32 microgram per ml of antibiotic. Then 16 microgram, 18 microgram, 8 microgram, 4 microgram, 2 microgram, 1 microgram, and there is no antibiotic in the control. Okay, so to each test tube we are adding the specific test microorganism or standard microorganisms, then allow for incubation. After incubation, we are testing the turbidity of each test tube. So the control, there is no antibiotic, there is maximum growth of microorganism, and the test tube containing concentration 1 microgram, 2 microgram and 4 microgram, there is the antibiotic concentration is very much low and there is no inhibition of bacteria. So that is indicated by the turbidity of these 4 test tubes. Okay, but in case of test tube containing 8 microgram, 16 microgram, 32 microgram and 64 microgram, there is no visual observance of turbidity. Okay, but if there is any chance of microbial growth which is not be presented as turbid, to test the presence of microorganism or growth of minute concentration, minute amount of microorganism in the test tube, we are transferring the contents of these 4 test tubes into 4 agar plates. Clear? We are transferring the contents of these 4 test tubes to 4 agar plates. Again it is incubated. After incubation, the 16 and 18 8 microgram containing test tubes or agar plates have growth. So which indicate in these two concentration there is the growth of microorganism. But in the concentration of 32 there is no growth of microorganism. Okay. So the MIC or minimum inhibitory concentration is 8. That is, it in the concentration of 8 microgram per ml, it inhibits the growth of microorganisms. So we can have the turbidity in this test tube. But the minimum bactericidal concentration, that is the concentration which kill the microorganism, is 32. That is at 32 microgram per ml which kill all the bacteria. So the minimum bactericidal concentration is 32 and minimum inhibitory concentration is 8. Clear? So that is the difference between MIC and MBC. Okay. Then in a case of agar dilution test, we are preparing the serial dilution in agar medium. The different agar test tubes containing liquid agar medium, to which we are adding different concentration of antibiotic and the test standard microorganism. Okay, then we are pouring the test tube containing agar medium into the petri plate and allow for incubation. After incubation, we are observing the growth or colonies of microorganism in the agar plate. Okay, clear. So the advantage of agar dilution method, each plate can be inoculated with the multiple bacterial strains. Clear. This slide also explains the difference between MIC and MBC. Okay, so in this there is no These are the different test solutions, different concentration of test solution, and which the turbidity indicate the growth, and the absence of turbidity indicate the absence of growth or the antibiotic activity. Okay. The second method used for antibiotic assay is diffusion method. The second method is diffusion method. In diffusion method, the agar plates are prepared by pouring nutrient agar medium into petri plates and inoculate with test organism. First, prepare the agar plates by pouring nutrient agar medium into the petri plates. Then inoculate the medium, that is inoculating the medium with the test organism, that is which is mixed with the agar before pouring or applied to surface after solidification of agar medium. So the first step is preparation of agar plates. Second step is inoculation of medium with the test organism. Then the plates are kept at 37 degrees Celsius for 30 minutes for the solidification of agar medium. Then the test antibiotic solution is applied on the solid medium by making cavities or by using filter papers or disc. The third step is applying antibiotic solution to the solid medium by making cavities or by using filter paper disc, that is by making cavities or by making filter papers or discs which are previously dipped in the test antibiotic solution. Then the plates are incubated, uh, during which the antibiotic solution diffuses from into the medium and inhibit the growth of microorganism. That is, the plates are then incubated, and during the incubation the antibiotic solutions which are present in the disc or which are present in the cavities are diffuses through the agar medium, and which kill or inhibit the growth of microorganism around the area in which the antibiotic solution is diffuses. Then after the incubation, the inhibition zones are developed. Okay, the inhibition zones are developed which can be seen around each filter paper or disc or each cavity. The zone of inhibition is seen around the cavities or it is seen around the filter paper or disc. The inhibition zones develops only if the test organism is susceptible to the antibiotic. That is, the zone of inhibition is developed only if the test organism is susceptible to the antibiotic. Then the sensitivity of the test organism to the antibiotic is determined by measuring the diameter of the zone of inhibition. Then sensitivity of the antibiotic is determined by measuring the zone of area of the zone of inhibition. For reading the zone of inhibition, we are using the zone reader, the antibiotic zone reader for reading the diffusion area. That is, the plates are incubated and allowing antibiotic to diffuse into the medium and inhibit the test organism growth. After incubation, the circular zone of inhibition are developed around each filter paper or disc or cavity. Then the diameter of the zone was measured by using antibiotic zone reader, and the larger zones indicated greater antibiotic potency and microbial susceptibility. The larger zone indicate the greater antibiotic activity and the lower zones indicate the lesser antibiotic activity. Now we are considering about the official methods or official assay methods for the determination of antibiotic potency. That is the standardized protocols which are used for accurate antibiotic potency determination, and these methods require careful attention to standard preparation, test condition and validation protocols. That is the official assay methods which are mentioned in pharmacopoeias, which are used for the accurate measurement of antibiotic potency, and these require careful attention to standard preparation, test condition and validation protocols. The standard preparation and units of activity. A standard preparation is a reference sample of antibiotic which have precisely determined potency expressed in international units or microgram per ml of pure antibiotic. These standard materials serve as the benchmark for all potency calculations in microbiological assays. That is, the standard preparations are the preparations which are used as reference sample of antibiotic assay. Okay, so example for standard preparations include for different antibiotic solutions, including for the assay of amphotericin B, we are using standard preparation of amphotericin B of 0.001064 unit. Okay, then for ampicillin, we are using a standard preparation of ampicillin which contain 0.001174 milligram in 1 unit, and for bacitracin we are using standard zinc bacitracin preparation which contain 0.01351 milligram in 1 unit, and for chloramphenicol we are using chloramphenicol of 0.001 milligram in 1 unit, and for nystatin we are using nystatin 0.000333 milligram in 1 unit. That is, the standard preparations are reference samples of antibiotic which are used for determining the potency of test antibiotic solution. The second official procedure is preparation of test sample. The test samples are prepared based on the pharmacopoeial methods. The first step, assay assumed potency. That is for determine the presumed potency per weight unit or volume based on available information about the test substance. That is, we are assigning the assumed potency of the test substance based on the available information about the test substance. Then preparing stock solution of test substance used for assay, and we are preparing the stock solution on the same day of assay or the day before the assay, and we are keeping the stock solution in refrigerator for further use. Okay, then the final sample dilutions are prepared from the prepared stock solution. Then third step is creating test dilution. That is, we are preparing different test dilutions of different concentrations which are assumed equal to median level of standard for 5 level assays. We are preparing standard solution according to pharmacopoeial conditions, and we are preparing the test dilution which are be in the median level of standard concentration. Then the next step is selection of test organism and inoculation. The test organism represent a carefully selected microbial strain which is sensitive to the antibiotic being evaluated. That is, the test organism selected must be sensitive to the test antibiotic solution, and the inoculum refers to the volume of microbial suspension that produces the optimal response when tested against the specific antibiotic. That is, the concentration of inoculum which refers to the volume of microbial suspension that produces optimum response when tested against the specific antibiotic. This optimal volume is determined through preliminary experimental trials and must be precisely replicated in each assay to ensure consistent and reproducible results. Okay. The next one is preparation of inoculum. The preparation of inoculum includes several steps. First one, maintain the culture. The test maintain the test microbe on slant of medium A and transfer to a fresh slant once a week. This mother culture is used as the source for preparation of inoculum. The second step is initial incubation, that is incubate the slant at specified temperature for 24 hours. Then surface transfer, that is using 3 ml of slant solution, wash microbes from agar slant onto rocks bottle containing 250 ml agar medium. And the next one is growth phase, that is incubating for 24 hours at a required temperature to achieve optimal microbial growth. The next one is assay media. Each antibiotic assay requires specific media. Formulations optimized for microbial growth and antibiotic diffusion. Media composition, pH and nutrient contents are precisely controlled to ensure reproducible results. The common media used for antibiotic assay including nutrient agar media, Muller-Hinton agar media, and specialized formulations designed by pharmacopoeial methods. These are certain examples of assay media which are used for antibiotic assay. The next requirement is apparatus and equipment. The apparatus and equipments are clean thoroughly to prevent contamination and ensure accurate result. That is, all the apparatus and equipments are cleaned thoroughly to prevent contamination. Then sterilization of the equipments and apparatus. This glasswares are sterilized by dry heat or autoclave or by steam sterilization. Then the petri dishes, glass, plastic or rectangular trays used for cylinder plate method application. That is, glass, plastic or rectangular trays are used as petri dishes for cylinder plate method application, and standard dimensions of 6 mm cylinders are used for making the wells. Then glass or plastic tubes are used as test tubes for turbidimetric methods. Okay, we are using petri dishes, test tubes and cylinders for making wells. Petri dishes may be glass, plastic or which may be rectangular or cylinder plates, and test tubes are also glass or plastic tubes. Then all the equipments are well cleaned for preventing the contamination, and all should be sterilized by using hot air oven or by using autoclave. Then the main part of antibiotic assay, that is the different methods, two primary methods used for antibiotic assay. We are already discussed about different methods of antibiotic assay, which including serial dilution method and diffusion method. And these two methods are the core concept or principle behind the antibiotic assay. The serial dilution method is also known as turbidimetric method, and the diffusion method is also known as couplet method. The serial dilution method is method B, that is turbidimetric method, and the diffusion method is method A, that is couplet method. The couplet method is also known as cylinder plate method, which utilizes solidified agar in petri plates with vertical cavities or cylinders for antibiotic diffusion testing. The method B is turbidimetric method. It is also called as tube assay methods, which measures microbial growth turbidity in liquid medium to determine antibiotic potency. we already discussed about different methods used for antibiotic assay and different conditions, different medium for antibiotic assay. We discussed that these are based on the pharmacopoeia standards. Okay, so specific antibiotic compounds require designated test organism, assay method, media types and incubation conditions as defined by official pharmacopoeia standards for the determination of its quantity and quality. Okay, this table contains the details of different methods used for the assay of different antibiotic solution. For example, in case of ampicillin, the microorganism used is Micrococcus luteus and the method is method A, that is diffusion method, and the medium used is medium D and temperature condition 32 to 35 degree Celsius. And for chloramphenicol, we are using E. coli as test organism and the method used is method B and the medium is C and temperature is 35 to 37 degrees Celsius. And for doxycycline, we are using Staphylococcus aureus as test organism and the method is B and medium is C at temperature 35 to 37 degrees Celsius. For rifampicin, the method is method A by using Bacillus subtilis and using the medium F and temperature 32 to 35 degrees Celsius. And for tetracycline, the organism is Bacillus cereus, method B and medium C at temperature 35 to 37 degree Celsius. So now we are considering about different antibiotic assay methods. The first method is method A, that is cup plate or cylinder plate method. This is a diffusion based technique which measures antimicrobial activity by observing the zone of inhibition around antibiotic containing cavities in solidified agar media. Principle of couplet method include the couplet method relies on diffusion principles. When an antibiotic solution is placed in a well or cylinder on a nutrient agar plate which is seeded with a test microorganism, it diffuses radially outward the agar matrix. That is, when an antibiotic solution is placed in a well or cylinder on a nutrient agar plate which is seeded with a test microorganism, it diffuses radially outward through the agar matrix from the well. The microbial growth is inhibited in the regions where antibiotic concentration exceeds the minimum inhibitory concentration, which creating a visible zone of inhibition. The zone diameter correlates directly with antibiotic potency and effectiveness. Okay, the microbial growth is inhibited in region where antibiotic concentration is more than the minimum inhibitory concentration, and here it creates a visible zone of inhibition, and the diameter of the zone of inhibition which is directly proportional to antibiotic potency and. So this is the method of doing the antimicrobial assay in couplet method. The first step, prepare suspension or culture of test microorganism is inoculated to liquefied solid assay medium at 43 to 45 degrees Celsius, or first adding the assay medium into the Petri plate and allowed to solidify, then add the inoculum to the surface of the agar medium by spread plate technique. Okay, then the mixture is poured to the empty sterile Petri plate to give uniform solid depth of 3 to 4 millimeter. Then place a uniform sized sterile metal cylinder or make cavity in solid media. By using sterile metal cylinder, make cavities of 6 to 8 millimeter diameter in the solid media. Then add equal volume of standard solutions and test solution of antibiotic to fill the cavity or cylinder. Then leave the plates for 1 to 4 hours at room temperature for the diffusion, for the proper diffusion of the antibiotic, and they incubate for specified time at a specified temperature, that is 18 to 24 hours at 32 to 37 degree Celsius for promoting the growth of inoculum. Then accurately measure the diameters or areas of the circular inhibition zone and calculate the results using equation or by graphically. Clear. These are the steps used for couplet method. First prepare the agar medium. Then pour the agar medium into the agar plates or petri plates. Then pour the inoculum to the surface of the agar medium. Or prepare agar medium and the inoculum to the liquid agar medium and mix it well. Then pour to the petri plate and allow for solidification. Okay. Then place a uniform sized sterile metal cylinder or make cavities in the solid media of 6 to 8 millimeter in diameter. Then add equal volume of test solution and standard solution to each cavity. Then leave for 1 to 4 hours for the proper diffusion of the antibiotic solution. Then incubate the medium or the plates at the specified temperature, that is 18 to 24 hours at 32 to 37 degrees Celsius. Then measure the zone of inhibition, diameter of the zone of inhibition, and calculate the results using equation or by graphical methods. So, this is the interpretation of zone of inhibition. The large zone of inhibition which indicate the significant antimicrobial activity against the microorganism. Then the small zone of inhibition which suggests either reduced antibiotic potency or developing microbial resistance. And no zone of inhibition indicate complete resistance or unaffected by the antibiotic. The microorganism is unaffected by the tested antibiotic concentration. Okay. So this is the interpretation of zone of inhibition. That is, the large zone indicate the maximum activity or potency. Then small zone indicate the lower activity or resistance, and the no zone indicates there is no activity or there is complete resistance by the tested organism. Then for the determination of the concentration of test antibiotic solution, a standard curve is plotted by plotting the concentration against zone of inhibition. That is, the diameter of zone of inhibition in the y-axis and the low concentration of antibiotics in the x-axis. Plotting a graph by plotting low concentration of standard solution in x-axis and the zone of inhibition, diameter of zone of inhibition in the y-axis, we get a straight line, and from the straight line we can calculate the concentration of unknown antibiotic. Then the next method is turbidimetric method or tube assay method. The next method is turbidimetric method or tube assay method. The principle involved in the turbidimetric method or tube assay method depend on the inhibition of growth of a microbial cell in a liquid culture media. That is, the principle depend on the inhibition of growth of a microbial cell in a liquid culture media. If the antibiotic kill the microorganism, the turbidity of the culture media reduces, and this was observed spectrophotometrically by measuring the absorbance of light. Okay, if the antibiotic kill or inhibit the growth of microorganism, which reduces the turbidity of the culture media, and it is determined by spectrophotometrically measuring the absorbance of light, and the absorbance is then correlated with the concentration determination of unknown antibiotic. Okay, then the absorbance is then correlated with the concentration determination of the unknown antibiotic. Then so this is the principle behind the turbidimetric assay. The principle is the inhibition of growth of microbial cell in a liquid culture media. That is, when antibiotic kill or inhibit the growth of microorganism, the turbidity of the culture media decreases proportionally. So we are measuring the absorbance of the turbidity but at a different wavelengths. So this is the steps involved in the determination of turbidity or turbidimetric method. First, prepare 5 different concentration of the standard solution by diluting the stock solution. From the stock solution, prepare 5 different concentration of standard solutions, and a median concentration of standard is selected as dilute the solution of the substance being exempt, that is unknown, to obtain approximately this concentration. That is, from the unknown solution, we are preparing a median concentration of the standard as the unknown solution. Then place 1 ml of each concentration of the standard and the sample in each test tubes in duplicates. Then to each test tube add 9 ml of nutrient medium which is previously seeded with the appropriate test organism. Then place in an incubator or water bath and maintain them at 37 degree for 3 to 4 hours. Then after incubation, add 0.5 ml of dilute formaldehyde solution to each tube to kill the remaining organism. Then measure the growth of the test organism by determining the absorbance at about 530 nanometer of each of the solution in the tubes against blank. Okay, so this is the procedure for turbidimetric assay. First, prepare different concentration of standard, then prepare test solution, then place 1 ml of standard solutions and test solution to each of the test tubes. Then to each test tube add 9 ml of nutrient broth which is already seeded with the test microorganism. Then place in incubator at 37 degrees Celsius for 3 to 4 hours. Then after incubation, add 0.5 ml formaldehyde to inhibit the further growth of microorganism. Then measure the absorbance at 530 nanometer. So this is the antibiotic turbidimetric assay result. If the concentration is no drug is added, then there is maximum growth of microorganism or maximum turbidity. If the concentration is more, there is no growth of microorganism or there is absence of turbidity. Then we are subculturing the test solutions into the solid agar medium. Then we are observing the growth by formation of colonies in the agar medium. Then if there is no turbidity or no growth of organism, then the test solution containing that test tube, there is no growth, no formation of colonies, which indicate the complete bactericidal activity of the test antibiotic. So then we are determining the unknown concentration by plotting the amount of growth or the concentration of the test solution or standard solution in x-axis and the absorbance in y-axis. Okay, from this we can calculate the unknown concentration. Then the advantage and limitations of turbidimetric methods, including in case of turbidimetric method, the advantage is there is shorter incubation period, that is 3 to 4 hours required for turbidimetric assay. But in case couplet method, we require 18 to 24 hours for the result. Then we are using automated spectrophotometric readings which reduces human error and higher throughput for multiple samples. Then disadvantage is the solvent residues or inhibitory substances interfere with the results, are not suitable for cloudy or turbid antibiotic preparations and require specialized spectrophotometry equipment. These are the disadvantages of methods. Then the key difference between coupled method and turbidimetric method including in coupled method we are using solid agar medium, but in case of turbidimetric method we are using liquid culture medium. In coupled method we are using longer incubation period, and in case of turbidimetry we are using shorter incubation period of 3 to 4 hours. And in coupled method visual zone measurement is doing, and in turbidimetry we are measuring by spectrophotometric reading. And coupled method is less sensitive to impurities, and turbidimetric method is more sensitive to impurity as we are measuring the turbidity by spectrophotometric method. And in coupled method clear preparations only used, and in turbidimetric method we are using